I'm Elenia Cinelli, and one field of my research activity is focused to study neural mechanisms underlying respiratory rhythm generation in the lamprey. Breathing is a complex behavior that involves rhythm-generating networks. Our studies investigate the main characteristics of respiratory rhythm generation in vertebrates and, in particular, the role of neural mechanisms involved in the neuromodulation of the lamprey respiration. The lamprey that is shown in this figure is a jellyfish that diverged from the main vertebrate line around 560 million years ago, and its central nervous system can be regarded as a vertebrate prototype with the experimental advantage that it has fewer neurons than higher vertebrates. In fact, the lamprey proved to be highly useful to identify neural circuits underlying rhythmic motor behaviors such as locomotion and respiration. Importantly, the isolated brainstem of the adult lamprey spontaneously generates respiratory rhythm activity in vitro. The respiratory activity that is shown in these original traces can be recorded from respiratory motor neurons located within the vagal nucleus. The neuronal aggregate responsible for respiratory rhythm generation is located in a region rostrolateral to the trigeminal motor nucleus and called by our research group the paratrigeminal respiratory group, the PTRG. The PTRG shows many similarities with the Prebetzinger complex that corresponds to the mammalian inspiratory central pattern generator. Several results obtained in these years support the notion that the PTRG is necessary and sufficient for the generation of respiratory activity in the lamprey and identify the structure as the respiratory central pattern generator. This cartoon illustrates some important connection and relevant neurotransmitters that influence both the PTRG and the respiratory motor neuron regions. Excitatory and inhibitory amino acids have a prominent role in the lamprey neuronal network. The PTRG is specifically sensitive to opioids, a characteristic reported also for the Prebetzinger complex. In addition, substance P and acetylcholine have a rhythmogenetic role. Recently, we have shown that the PTRG is sensitive to exogenous ATP with a biphasic response, and astrocytes there located strongly contribute to the modulation respiratory pattern. These results emphasize that some features of neuronal mechanism of respiration are shared between lamprey and mammals and maintained throughout evolution, thus making the lamprey a useful model to advance knowledge into the control of breathing. I would like to acknowledge my collaborator in Florence and our collaborators in Stockholm at the Karolinska Institute, and thank you all for the attention.